if indeed in the fourth line setting, we're seeing efficacy similar to what we were seeing in second line sunitinib, what is the role of, or what is the potential role for ripretinib in earlier line treatment, and how is that currently being studied?
So yes, it is being studied, but let me back up a step. I think you very nicely outlined the mechanism of action. And at least personally from my standpoint, but I'll see what your thoughts are. I think one of the most interesting aspects of ripretinib, even long before we saw some encouraging clinical data, was indeed its mechanism of action. That is this switch pocket inhibition. To very much simplify it, sort of tells me that if it's going to minimize or delay the conformational changes in the kinase and delay the development of secondary resistance, the drug may very much have a role in earlier line settings, right? So from a mechanistic standpoint, it sort of kind of made sense. The PFS data in the fourth line setting certainly support that notion. And then the final part of that equation is that we've already sort of outlined that sunitinib, while a very important addition to the therapeutic armamentarium, has been a part of standard of care, is probably going to remain a standard of care. I think there are things that can be made better. I think a better therapeutic index with improvement in patient-related outcomes could certainly be a very helpful addition to the current therapeutic armamentarium. So for all of these reasons, theoretically, I think it absolutely makes sense that ripretinib gets tested in the earlier line setting. And I think as you outlined, there is an INTRIGUE trial going on. It's a randomized phase 3 trial comparing ripretinib to sunitinib in the second line setting. The trial is ongoing. I think we await the results of that trial. And the hope clearly is that despite the comparison to an active control, and here is where the glitch comes around, right? I think in the soft tissue sarcoma space, we've seen plenty of trials with active treatment control fail and some of them succeed. So I think the INTRIGUE trial that will randomly compare ripretinib to sunitinib in the second line setting will really truly answer the question as to the value of the mechanism of action as it relates to delaying the secondary resistance development. And then frankly, the patient's quality of life, how long will they continue therapy and that sort of stuff. I think if we go back to the Invictus trial, I think a testament to the tolerability of the drug is still relatively low discontinuation rate and relatively low and manageable dose interruptions and dose adjustments. And I think these are parameters that clearly tell us how well the drug is tolerated. I think not just this classic phrase, drug is well tolerated, but those are the actual parameters that sort of reinforce that.